Welcome to the fascinating world inside our cells, where a delicate balance act keeps everything running smoothly, a process known as proteostasis. Think of it as the cell's version of a highly efficient factory, constantly managing proteins to ensure they fold correctly, function properly, and are recycled when they're no longer needed. One of the key players in this process is the endoplasmic reticulum, or ER. It's like the cell's protein factory, where proteins are assembled and folded. But sometimes proteins misfold. That's like a factory producing defective parts. When misfolded proteins start accumulating, trouble brews, leading to stress in the cell called oxidative stress, which can damage mitochondria, the cell's power plants, and even reach other structures like peroxisomes that handle reactive species, those pesky reactive oxygen and nitrogen molecules. To fight this chaos, cells have a quality control system called the unfolded protein response. This is the factory's emergency response team. When misfolded proteins pile up, sensors in the ER detect the problem. Once the sensor is an endoplasmic reticulum kinase, specifically anisotro-required kinase 1, or RE1. RE1 acts like a transmembrane kinase embedded in the ER membrane. When activated, it phosphorylates itself and then activates transcription factors like Xbox binding protein 1, which travels to the nucleus to turn on genes that help clear out misfolded proteins. But RE1 isn't alone. There's also a protein kinase RNA, like ER kinase or PERK, which upon activation phosphorylates a key molecule that attenuates translation, slowing down protein production to prevent further overload. This is like putting a temporary halt on the assembly line until the backlog is cleared. Meanwhile, another ER sensor activating transcription factor 6 moves to the Golgi apparatus, where it's processed into an active form that enters the nucleus to boost the cell's defense and repair mechanism. All these responses work together within a complex network called the unfolded protein response, ensuring misfolded proteins are refolded or degraded, primarily via associated degradation pathways or autophagy, the cell's recycling system. Autophagosomes form as double membrane structures that engulf damaged proteins and organelles, including mitochondria, and deliver them to lysosomes for their disposal. Speaking of recycling, cells also use autophagy to manage cellular stress and maintain homeostasis, especially during embryonic development or neuronal precursor stages when cell growth and differentiation are in full swing. Autophagy activity can be influenced by signaling pathways like the mammalian target of rapamycin, or mTOR, which acts as a cell growth regulator. When nutrients are abundant, mTOR complexes promote growth, transcription, and translation, but under stress it's suppressed to activate autophagy. Through all this, chaperone proteins like glucose regulated proteins act as molecular helpers, assisting in proper protein folding. They're like the factory's quality inspectors, making sure products are up to standard before they leave the assembly line. So in essence, our cells are like bustling cities with a sophisticated intricate network of factories, PERKs, and recycling centers, all working silently behind the scenes to keep us healthy, sharp, and resilient. Next time you think about what keeps your cells functioning smoothly, remember the incredible choreography of proteostasis, the unfolded protein response, nature's own maintenance team.